So with that, we come to the official part. So Professor Gruber, before studying, he also got a diploma in informatics and then received his doctor, or his title of doctor, in 2007 at the famous University of Constance. In 2007, then he became the professor of experimental psychology at the University of Osnabrück. His research interest mostly centers around visual perception, brain oscillations, and especially today's topic, the role of Gammaband oscillations in processes like binding or other cognitive procedures. In his last work, or in the last work, his work has also shifted towards the field of VR research, where his team focuses on increasing the ecological validity of psychological studies. And besides his papers, he's also the author of the book Gedächtnis. So, what is this about? In day-to-day life, we really never question why we perceive objects as they are. So why we perceive the table in the same in the color it should be, and not mix up the perception of maybe the screen you're looking at and this color of the table. But how does our brain actually manage that? And this is where Gammaband oscillations come in, and that's where I would like to hand over to you, Professor Gruber.
Sebastian, Aska, thank you very much for organizing this evening. Thank you so much for having me here tonight, and thank you for the kind introduction. So let me get right into it. So the intention of my talk this evening is I want to show you, since you're all young researchers, you might be interested, that research is not always a straightforward success story, but it can be marked by setbacks and also disappointments. And that's why I've titled my talk Ups and Downs of a Research Field. In particular, I want to talk about Gammaband oscillations and what ups and downs this field was characterized by. So here you have a brief overview of my talk so you know where you're going. First, I want to talk about a long-held belief, namely that induced Gammaband responses in the human EEG represent object representations and memory. So this is an EEG talk tonight. And the first part is the up part of the story, the ups. The second part then, this is the down part. Here I say 2008, there was a publication that claimed this Gammaband, this famous Gammaband is nothing but eye movements. So I want to talk about the downs of this research field. And in the second half of my talk, I want to show you some possible solutions to this problem to draw some conclusions at the end. So this is my overview. And before I go right into it, just a brief questions. I would assume you don't see my mouse pointer, don't you? So or do you
see it? At the moment, we
see. We see it.
You see my mouse pointer so I can point? That's good. Good to know. So then let's start. Induced Gammaband responses in the human EEG. Sebastian Wilhelm has explained it all already a bit what it is all about. So this is a macaque brain, what you see here, and the macaque brain, like the human brain, is characterized in the visual system, but also in all other systems by many, many areas. Here you see a figure from a publication from Velleman and Van Essen, over 30 specific areas in the visual cortex of the macaque. You have areas here named MT, V4, V5. And all these areas process different aspects of a stimulus. So we know, for example, that color is processed in an area called V4. This is here in the brain approximately. Motion, the direction of motion, is processed in an area called V5. Certain forms of an object are coded in anterior parts of the inferior temporal cortex. So everything is coded in different areas of the brain. There is not a single neuron which codes a whole stimulus. There is no grandmother cell, or like in Sebastian's example, the unicorn cell, which codes for the whole unicorn he has shown us. So this poses a problem. How does the brain bind together all these activity patterns in different great areas, and this is the so-called binding problem in neuroscience. And one solution to the binding problem is shown here on the next slide. It was brought up by many researchers, Hebb, a Canadian psychologist, already in 1949, but also from neuroinformatics and from animal recordings. And the idea is the following. If you look at this giraffe, then the idea is that this giraffe is not coded in a single neuron, but in a distributed assembly of cells, a cell assembly. And this cell assembly is bound together by synchrony. The cells that code for the giraffe here, they fire in synchrony. So here, to read out this final statement here, which speaks for that synchrony selectively takes the responses of neurons that code for the same object and demarcates their responses from those neurons activated by other objects. And usually on this slide, the red dots here, which stand for the giraffe, they should blink in synchrony. Now they don't. So I need your imagination now, because the PowerPoint doesn't blink, but it doesn't matter. You can imagine that the red dots stand for neurons coding for the giraffe, and they all fire in synchrony in one pattern, yeah, all together. And that's how they are bound together. That's the basic idea. And if we look at this synchronous firing of action potentials at a macroscopic level, with macroscopic level, I mean something like the local field potential or the EEG or the MEG, then the synchronization is mirrored in the form of oscillatory activity. And here you see such an oscillation in an EEG. On the left, a person with an EEG cap on, and then you see an oscillation in a time by amplitude plot. Here in this time range, you told me you see my mouse pointer, this is an oscillation. That's a signature of this binding process. And this oscillation does not occur in any frequency, but here this red square indicates an oscillation in a frequency range from around 25 to 100 Hertz. This is the so-called gamma band. The frequency bands in the EEG, they are named by Greek characters. The most famous band in the EEG is the alpha band, discovered by Belger already in the 1920s, around 10 Hertz. And then there's a theta band and the beta band and the gamma band. This binding band lies between 25 and 100 Hertz. Now, one usually does not look at these oscillations in the form of these time by amplitude plots here, but you usually, in this time by amplitude plot, you would need to count how many cycles this oscillation have in order to say it's a gamma band. We don't want to do that. That's why we calculate a frequency transform, and the result of a frequency transform is a spectrogram. And here on the x-axis, you don't see time anymore, but frequency, amplitude still on the y-axis. So now you have one peak at 40 Hertz, which depicts this oscillation here. Another form of frequency transform, which is used very often in neuroscience, is a time by frequency analysis. This gives you one dimension more in the result. This is a time by frequency plot of this oscillation here. And what you see here is, or let me put it in another way, you don't need to understand how this is calculated. I will say more to that later, but for now it's only important that you understand how to read these time by amplitude plots. On the x-axis, you still have time. On the y-axis, you have frequency now, and the amplitude of a frequency is color-coded. So this red blob here stands for the oscillation in the time domain you see here in the upper left panel. So now you have a time by frequency plot, which gives you the frequency of an oscillation, when the oscillation happens, and how strong the oscillation is color-coded. And one more thing you need to know from a methods perspective to understand what I'm going to say is that you can differentiate between oscillations which are evoked or induced. So there are evoked and induced gamma oscillations. An evoked oscillation is the following. If you look at this figure here, then you see a time by amplitude plot of an oscillation in a number of exemplary trials. And around 100 milliseconds, here right after stimulus onset at 0, you see an evoked oscillation. You see that this oscillation looks exactly the same in each experimental trial. It's time and phase locked to stimulus onset. An induced oscillation is different. You see this around 300 milliseconds here. You see this jitter in latency from one trial to the next. This is an induced oscillation. If you now average these signals from single trials in the time domain, what happens is the following. The average shows you the evoked oscillation. That is like an event-related potential, a conventional ERP in EEG response. So the evoked gamma band response is clearly visible, but the induced, due to this jitter in latency, that averages out. And if you now do a time by frequency transform, you look where are the red blobs, so to speak, then you see the evoked blob, but the induced is not visible. So one usually does in this research field, if you look at oscillations in the human brain, you don't average in the time domain, but you transform each single trial into the frequency domain. And then next, you average the signal not in the time domain, but in the frequency domain. You average these time by frequency plots. And when you do that, then you still have the evoked response here at 100 and the induced response. So very important that you keep in mind an induced response occurs with a jitter in latency, and you have to analyze the data in a way that you transform each single trial into the frequency domain and average into frequency domain. So keep that in mind for later, for the remainder of my talk, please. But now I first want to show you some exemplary findings, and I want to focus on this induced gamma band response. This is now not only findings by my group, but by others as well. Here, one very famous study is by a French group around Catherine Talon-Bautry. She has presented her participants figures like you see here on the left panel, either Kanisha figures, which form a triangle. You see this triangle illusion here, or the Pac-Man, which induced this illusion were rotated. So on the right side here, when you see the triangle, you have to bind something in order to see a triangle. Not so when the Pac-Man are rotated. And then in the results, if you look at the time by frequency plots, you clearly see at around 300 milliseconds an induced gamma blob for the triangle, but not so, and now you look please to the left side here, not so in the control condition where there's no triangle. A similar result was found by Rodriguez and colleagues, published in Nature. He showed either pictures of faces, as you see here on the right side. This is a face. If you look closely, you might see this face, or a control condition. If you rotate these black and white drawings, it's sensory, the same sensory information, but no face visible. And then you clearly see here this induced gamma peak, possibly related to the binding of the features of a face in the phase condition, but not in the control condition. And one final study by our own group, we presented our participants pictures of familiar or unfamiliar objects. Here the sunflower or such an unfamiliar thing on the left side. And clearly you have to bind information in order to see the sunflower. You have to activate your sunflower network. And when we then look at at the frequency response, we see a nice induced gamma peak at 300 milliseconds, which is, if we subdivide it according to conditions, if you now look at this line plot, which is higher for familiar as opposed to unfamiliar. So to draw a first conclusion, this induced gamma band response around 200 to 400 milliseconds shows an increase whenever different features have to be bound in order to activate a so-called cortical object representation of a stimulus. And this is the so-called representational hypothesis made famous by Catherine Talon-Baudry and her co-worker Olivier Bertrand, a very famous paper in 1999, already an old paper, but this representational hypothesis is still a thing, so to speak. So let's continue. The question now is, are these cortical object representations, are they fixed entities, or can they be changed, manipulated by psychological manipulations? The hypothesis is, I think, quite straightforward. These object representations must be modulated by learning and memory in order to allow for adaptive behavior. I think that's clear. You can't have fixed object representations. If you learn something, they must change. If you recall from memory, they must change. So something has to happen here. So to test this hypothesis, we used an implicit memory paradigm, namely a repetition priming paradigm. Repetition priming is just as the name says, repeated presentation of a stimulus. And usually you have some improvement in processing if you see a stimulus for the second time. So, for example, you see pictures of animals, and whenever you see a giraffe, you have to press a button, and we measure the reaction time. When we see the giraffe or the target animal for the second time, you're usually faster. This is priming, repetition priming. But this is very simple, a very simple memory paradigm. Basically, the most simple memory paradigm one can conduct, and that was also the idea of this experiment, of this first experiment. Keep it simple. If you start a new research field, you better keep it simple at the start. So we repeated pictures of, now comes the paradigm, we repeated pictures of familiar objects. Here, for example, this giraffe was presented twice, and as a control, we repeated pictures of unfamiliar objects. So repetition of familiar and unfamiliar object here indicated again in red and green. So what have we found? Here you see the time by frequency plots of the four conditions. First, presentation. If you first look at the left side here, the first presentation, we find a nice induced gamma band response at 300 milliseconds for familiar objects, but not for unfamiliar objects. This is a replication of the familiar-unfamiliar effect, binding for objects, no binding for no object. But if we repeat the familiar object, the gamma band, and now you have to look into this direction. Please look to the plots on the right side now. You see for the familiar repetition, you see a decrease in gamma band responses. Very different finding for unfamiliar object. Here you see unfamiliar objects. Here you see a clear increase in the gamma band response if you repeat the unfamiliar line drawing. And just as a side note, if we look at the conventional event-related potential with its components P1, N1, P2, you name it, you don't see such a differentiation. You just see a repetition suppression. That's a proper term for the suppression. When you repeat something, you see a repetition suppression for familiar, unfamiliar. You don't see this effect of suppression and increase for unfamiliar. So how can we interpret these findings? As follows. If you repeat a familiar object, then for the second presentation, your network must be more efficient because you're faster. We see priming effects. So somehow this network must become more efficient. And one way to make a network more efficient is just not to activate the whole network. If you see a giraffe for the second time, you don't need the color of a giraffe or the information that it has 4 legs or whatever. Yeah, whatever. You don't need everything to say it's a giraffe. So this network is sharpened. That's the term for this process, which happens, a sharpening of a network. And this sharpening is reflected in a decrease in gamma band responses. However, or in contrast, when you repeat an unfamiliar object, there is nothing to to sharpen in this for these unfamiliar objects. So a new cell assembly has to be established. That's the formation of a cell assembly, and that's what you see then as an increase in the gamma band response. So what we can, to draw a brief interim conclusion, these induced gamma band responses are a valuable neural index to study implicit memory, this simple, very simple form of memory. So we asked now the next question: if it works with implicit memory, what about explicit encoding and retrieval? So I want to show you briefly two experiments on explicit memory. Encoding. Let's start with the encoding phase of an experiment. You know that memory, usually you have three phases. The encoding, that's when you learn something new. Then you have a consolidation. Maybe the network is consolidated while sleeping or whatever. And at one point you have to recall the memory. That's retrieval. Now we start with encoding. What one usually does to study encoding is one presents words or pictures. It doesn't matter if you take words, pictures, or whatever. And for every word you show during a study phase, you record the EEG. Cat, EEG is recorded. Trouser, EEG is recorded, and so on and so forth. Then, after a while, there's a retrieval phase. This a memory test. Have you seen the word cat before? Have you seen the word bird before? So this retrieval or this memory part is usually not announced in advance. Usually you have an incidental learning paradigm, but it's not so important now. Important now is the following. If you see the word cat again in the retrieval phase and the participant answers, yes, I remember this item, then yeah, it's a remembered item and something had to happen before during encoding because it's now remembered. If our participant sees the word bird and says, no, I don't remember bird, then obviously the word bird, yeah, it was never presented. That is correct as well. That's a correct. CR stands for correct rejection. So that's correct as well. But if the participants, yes, now it's important, if the participant sees the word car and says, no, I have not seen the word car before, then it's forgotten because there was the word car. This is a forgotten item, a miss. And if we now want to learn something about encoding, we simply have to compare remembered items to forgotten items. So we encoding related, that's now the proper term is subsequent memory effect. We compare subsequently remembered to subsequently forgotten items. So something has to differentiate between car and cat because cat is remembered and car is not. And now, yeah, the hypothesis is quite clear. This should be something in the gamma band because this has to do with object representations, with networks. And in fact, yes, if we look at the gamma band response, now I have not a time by frequency plot, but to show you the final results, I took a line plot here of the gamma band response in the lower part of this figure. You see the gamma band peak around 300 milliseconds is higher for subsequently remembered as opposed to subsequently forgotten items. So gamma is related to encoding. In the upper part of this figure, you see the conventional event-related potential. And obviously there is a difference as well, because yeah, this encoding was studied with ERPs for a long time, yes, and there's a difference, but it's later. Gamma band differentiates earlier. So to conclude, gamma band induced gamma band responses are modulated before any effect in the ERP, and gamma band responses are an index of encoding of new stimuli. Again, the formation of a memory trace. That was encoding. Now let me say a few words on the retrieval. If we talk about the retrieval of information, we have to differentiate it between two sub-processes. So if you recall a stimulus from memory, your brain usually does two things. This two things are formulated in the so-called dual process account of recognition memory, and these two processes are familiarity and recollection. Now, what is familiarity and recollection? Familiarity refers to the subjective feeling that an item has been experienced before. As the name says, something is familiar. Recollection, in contrast, is the remembrance of prior events, additional episodic information you have with the stimulus. I have an example for you. Maybe this becomes clearer. So if you see this person, he might be familiar. Maybe you say, ah, yeah, I have seen this guy before, but there's no recollection. You can't recall any additional episodic information which you connect to this person. And now I show you another picture. Now you should have, maybe, I don't know if you have seen him before, but maybe you have seen this person before. It's a famous artist from Hamburg. And now you say, oh yeah, now I know it's Olivia Jones. Here on the left, Olivia was just familiar, but here on the right, I recall additional episodic information, and this is recollection. So this is a very important difference when we recall stimuli. We can either say something is old based on familiarity, or we can say something is old, I've seen it before, based on recollection of information. So now we want to check if induced Gammaband responses are rather related to familiarity or to recollection. And to do so, we used a so-called source memory paradigm, with the correct source being a trademark of recollection. A source, this is just the recollection, the remembrance of additional information related to a stimulus. I show you how this paradigm works. So during encoding, we presented pictures of objects, but now subjects had a task. No, they had two tasks, two different tasks. Either a cue told them, hey, with the next picture, you have to decide if this is bigger or smaller than a shoebox. So here for this pool table, the participant would say, yeah, it's bigger. Or with another cue, cue number 2, participants had to decide if this object is man-made or natural. So here they would say butterfly, this is natural. Or here, the lighthouse, this is man-made. This is encoding. But we are interested in retrieval. That's the next phase of the experiment, the phase during which we measured EEG. We have presented an item again, for example, the butterfly. And now we had two questions. Have you seen this picture before? And if the participant says now, yes, I've seen the butterfly during encoding, it's old. Then we had another question. What kind of classification did you make during encoding? Was that the shoebox task or the man-made natural task? And this targets this additional episodic information. If you can answer this question, you have recollection. If you just say, oh no, butterfly, yeah, it was there, but I don't know anything else regarding the butterfly, then it's just familiarity. So, and what have we found? Let me briefly talk you through this results slide. Again, a gamma band peak at 300 milliseconds, induced gamma band response with a nice posterior distribution. And what you can see here is that this gamma band response is much higher for stimuli which were classified as being old. Independent if the source judgment was correct, independent of the fact if participants were able to say, was this the man-made task or was this the shoebox task? Whenever something is, whenever something is familiar, we see an increase in gamma. So where is the recollection? It's not in the gamma band. There are also other oscillations in the brain, for example, theta band oscillations or theta band. Sorry, it's a talk I have to give in English. It's a theta band oscillation. This is a slow oscillation, not as fast as gamma, around 5 to 7.5 hertz. It's also a bit later. And this theta band oscillation, these oscillations, these forms of oscillatory activity, they are able to differentiate between correct and incorrect source judgments. So induced gamma is familiarity. Yes, I've seen it before. And then if you recall additional information, yeah, it was this and that task, or this person is not only familiar, this is my dentist, or whatever. That is then in the theta band. So we propose that gamma band responses are a signature of familiarity-related activity, whereas theta band oscillations are related to recollection. So I could stop my talk now. I could show you this little guy here, which if the animation would work, bends over and says, applause, applause, we found an important neural basis of mnemonic processing, namely gamma band responses. They reflect object representations and yeah, they go up and down, whatever you do with your memory. Yeah, if it would be so easy. But now I come to the down part of my talk. So let's start with the second half of my talk. In 2008, it was claimed that gamma band responses reflect eye movements. And now in the next part of this talk, I have to talk a bit about methods and signal processing. I hope this is not too much. I try to keep it simple, but it's important so you understand what I want to say. But first give you an intro. What happened in 2008? So, I mean, if you saw these results I've shown you so far, it's implicit memory, it's retrieval, it's encoding, everything. Everybody was quite excited about this gamma band responses. It was really a hot topic, so to speak. Everybody said, oh yeah, with gamma band responses, you can do a lot. You can really do brain research with this response. And then there came a publication, there came out a publication in 2008 by Yuval Greenberg, and they claimed, no, we don't have any gamma band responses in the human brain. This, what you measure, you EEG guys, this is just eye movements. It's miniature saccades, small eye movements. Miniature saccades are small, yeah, small jerk-like eye movements that occur during fixation. They're not big like major saccades if you look from the left to the right, but they're just 0.5 degrees or so, 1 degree, small saccades. And their functional role is usually to counteract receptor adaptation so that the percept you have doesn't fade. If you use specific drugs to counteract these miniature saccades, your receptor fades. You don't see anything anymore. So they are counteract receptor adaptation. They are also discussed to correct fixation errors. But for now, it's not so important what their functional role is. I want to show you what they do with the EEG. So here in this plot, you see such a miniature saccade measured with an eye tracker. So you have the eye position first at 0, and then you have a horizontal eye movement and a vertical eye movement, the vertical in red and the horizontal in blue. And if you use an eye tracker, you see here this sharp slope when you have the eye movements to the left or to the right, up and down, whatever. And if we now measure EEG during such a miniature saccade, MS abbreviated, I use the abbreviation MS from now on, then this MS is accompanied by a signal in the EEG. This is a so-called spike potential. This little guy. So if you look at the blue line here, the EEG, and I would not tell you that there is something, namely the spike potential, you might not see it. It's just ongoing EEG. There's nothing special. But now I told you, look, there's a spike potential which always occurs when there is an MS. Yeah, then you might see it. So now the next question is, how do these SPs influence the EEG signal in the frequency domain? And that now I have to take you in this brief excursion on digital signal processing. And I hope I have it as simple as possible to make it clear what I mean. Brief excursion. There's somewhere else now. Let's have a look at the characteristics of different signals in the frequency domain. For example, if you have a sine wave in the time domain, a really pure sine wave, and you do a frequency transform and you look at the spectrogram, then you just see one peak, a peak exactly at the frequency of this oscillation, in this case at 5 Hertz. And if you use our time by frequency transform, then you see an ongoing red blob at 5 Hertz. So this is straightforward. Sine wave, one peak in the spectrogram. Now let's look at a different signal, a square wave. What a frequency analysis wants to do is it wants to model this square wave by oscillations, by sine or cosine waves. How could we model the square wave? Okay, first we could say, let's take, Anna, now first please look at the lower left of this figure. You said you see my mouse pointer, so we can model this square wave. When you look at my mouse pointer, yeah, I can take a sine wave approximately with 3 peaks and troughs here, so to model this square wave. And then I add a little bit of a faster oscillation to this sine wave, and then my signal looks like that. Then I take this previous result and add another fast oscillation. By adding, I really mean add a simple plus operation. You add this and then it looks like this. Then you take this result and add another even faster oscillation. And you do that again. And now you see, the more fast oscillations you add, the more it looks like a square wave. So to model a square wave in the frequency domain, you have to add up a large set of sine waves of different frequencies, particularly of fast frequencies. And if you then look at the spectrogram of such a square wave, then obviously you see the following. You don't see in the spectrogram one peak, but a horizontal line, because you need all frequencies to model a square wave. So just let that sink in for a second. To model a square wave, you need all possible frequencies. Now, the next signal we could look at, that's a so-called Dirac pulse. A Dirac pulse is defined as a signal with infinite amplitude. So that's more a theoretical signal, but doesn't matter for now. It's a signal with an infinite amplitude at one point and 0 elsewhere. So just here, nothing happens, nothing happens, nothing, and then a pulse, and then nothing, nothing, nothing. How can we model this in the frequency domain? Basically like a square wave. We need all frequencies to model a Dirac pulse. And if we look at this Dirac pulse in a time by frequency transform, then you see the following. You see, here's your Dirac pulse, but you can only model it by adding all frequencies. All frequencies. It starts at 5 Hertz and 20 Hertz, 40, 60, 80. You need all frequencies to model this Dirac pulse. So now comes the problem. Now let's go back to our miniature saccades and our saccadic potential. This saccadic potential, it shares, that's the problem, properties with the Dirac pulse. Namely, it's a transient signal, very short-living, and it has sharp edges. So if we do a frequency transform of a saccadic potential, you see this candle-like thing here during the saccadic potential, and this looks, I hope, familiar to you now. This looks like the Gammaband responses I have shown you in so many studies. This is here this familiar, unfamiliar study, and this is the gamma response, and this is the Dirac pulse. This is the time by frequency transform of a saccadic potential. So in the frequency domain, saccadic potentials and Gammaband responses, they look similar. Problem number one. But there are further properties of MS and saccadic potentials which are problematic. This is a study from Engbert and Kliegel. They have looked at microsaccades after the onset of a stimulus. And for now, it's not important what the stimulus was and what the different conditions were in this study. What is important is they have measured microsaccades, and you see that the miniature or the microsaccades show a maximum at 300 milliseconds. Same time range as the induced gamma band response. Not a problem. So, and to make things worse, I have one more. If you look, each little dot here in this graph is one microsaccade, one miniature saccade, and each line is one experimental trial. So you see many miniature saccades here in this typical time window at 300 milliseconds. But if you have a closer look, then you see that they not always occur, this saccadic potentials at 257 milliseconds, but they jitter from one trial to the next. So these saccadic potentials are induced, like the gamma band responses. So gamma band responses are induced, occur at 300 milliseconds, and are characterized by this candle-like pattern in the frequency domain. Everything the same as saccadic potentials. They're also induced, they're also at 300 milliseconds. And yeah, now what Yuval Greenberg 2008 concluded was the following. Whereas neuronal gamma band oscillations were shown conclusively with other methods, for example, single cell recordings in animals, were conclusively with other methods, the broadband transient induced gamma band response recorded by scalp EEG reflects properties of miniature saccade dynamics rather than neuronal oscillations. So basically, we can say now, okay, that was the end of gamma band research. And if my animations would work, you would see a stone now falling on this little guy standing here. But again, I need your imagination. You have to imagine how this little guy who is happy on his gamma band response is squished by a stone. That was the end of gamma band research. Bad luck. So that's what was the down part of my talk. So now for the remainder of my time, I want to show you some. Yeah, okay, basically we could give up now. We could say, okay, it was nice to do studies in gamma band response. Now let's give up and study something else. That was it. And basically, yeah, many researchers have did so. So if you look at the publications in the gamma band field after 2008, the field was basically dead. There were hardly any publications anymore, although it was such a hot topic. It was such a highly researched field. There was a really, yeah, hardly any publications anymore. So that's one solution. Just do research on something else. What else can we do? So what are possible solutions to the problem? Yeah, I want to show you three: ignorance, alternative dependent variables, and methods to remove the artifact. Now first, ignorance. That's something that really happens. I have here a time by frequency plot from a publication which was published after 2008. I don't want to do some colleague bashing now. That's why I don't have a reference here. But some colleagues, they have just ignored the problem. They kept publishing these gamma band candles, these miniature saccade candles, and they ignored the problem. That happened, yeah. But as I said, I don't have any names here and I don't want to reference anything. But if you see a publication after 2008 and you see this candle, it's really hard. I mean, sometimes you ignore other findings. We have so many publications in the field, you ignore other findings. But this 2008 publication by Yuval Greenberg was really, really hard to ignore. You must be really blind not to ignore this publication. So yeah, but one solution, ignorance. Another solution, just use another dependent variable. So forget about Gammaband responses. There are many other indexes in the human EEG which can be used. For example, the steady state visual evoked potential. I want to mention that very briefly. So what you can do, if your main interest, your research interest, yes, is not the mechanism, this, if gamma, the synchronization is the mechanism, but if you're interested in the phenomenon, for example, priming or memory, you can obviously use another index in the EEG if the mechanism is not your main interest, but the result of your research. So, and one different variable is this steady state visual evoked potential to study the functional properties of cortical object representations. That's our topic. We can use steady state visual evoked potentials. What is a steady state? Steady state is the following. And now again, I need your imagination because my animation does not work. You see this guy here, and he looks at a light which goes on and off. Just imagine now, please, a blinking or a flickering light which goes on and off. And what your brain does when watching a flickering light, for example, at 10 Hertz, it follows this oscillation. So if a light flickers at 10 Hertz, your brain starts flickering at 10 Hertz. If a light flickers at 15 Hertz, your brain starts flickering at 15 Hertz. And the assumption is here that flickering stimuli act as a pacemaker for networks which are responsible for the processing of a stimulus. And this network activity is reflected in the steady state visual evoked potential, this oscillation. So in other words, I can trigger the giraffe network I've mentioned at the beginning of my talk. I can trigger it if I flicker the giraffe at 10 Hertz. Then the giraffe network starts in the brain starts flickering at 10 Hertz, and I can measure 10 Hertz response. So what we have done with this method was a work by Ulla Martens, my colleague Ulla Martens, and she said, okay, we have to check if sharpening and formation, the two mechanisms of repetition priming I've mentioned before, if they really exist. Do we also see these mechanisms, sharpening and formation, with another method? And to that end, she has presented pictures of familiar and unfamiliar objects as you have seen it before. She flickered them at 12 Hertz to elicit a steady state response. And what she found, or what she expected, was, yeah, we want to see sharpening. If you repeat familiar object, the cell assembly becomes more efficient. And we want to see formation, the establishment of a new assembly, if we repeat unfamiliar stimuli. And in fact, that happened. Now look at the lower part of this figure, not at the words given in red. You see that what we found is the steady state response, this ongoing oscillation due to the 12 Hertz, was increased when we repeat familiar stimuli, but it decreased when we repeat familiar, but it increases when we repeat unfamiliar. So sharpening and formation are safe. These mechanisms exist. Gammaband is not safe, but the memory-based mechanisms, they are safe, sharpening and formation. And so we have also done other steady state studies on explicit memory but I have no time to talk about that now, and it's not a steady state talk. In the last part of my talk, I want to talk about another approach to deal with this problem of the miniature saccades. And this is a method or an algorithm which we have proposed to remove this artifact, this saccadic potential. How can we remove this potential from the EEG so that we don't have the problem anymore? And this algorithm we have developed is called, or we we named it COSTRAP. COSTRAP stands for Correction of Saccade-Related Transient Potentials. This algorithm was developed by two great guys, two great colleagues of mine, Uwe Hassler and Nelson Trujillo Barreto. Nelson is a physicist, also always good to have a physicist on board if you deal with complicated signals. And the idea behind this approach is the following. So just to give you a brief state where we are now, I need your attention now. It's again a bit method loaded now, again for around 10 minutes, so that you know how long I need your concentration. 10 more minutes, please. So these saccadic potentials, you can't detect them by conventional methods in the EEG. By conventional methods, I mean, for example, if you have done EEG before and you want to detect blinks, then you can say, okay, a blink has 100 microvolt and everything above 100 microvolt is kicked out of my EEG and the EEG is fine. But these saccadic potentials they're too small in amplitude. You can't use an amplitude threshold because then you would throw away your whole EEG. And it wouldn't solve the problem if you would detect them by an amplitude threshold, because there are simply too many of them. You would delete your whole EEG recording. So one idea is to use an approach to remove these artifacts, and this approach is called independent component analysis. I don't want to go into detail of independent component analysis. I just want to explain you the principle. Basically, what you do if you want to apply it and you have any tool to analyze your EEG, you usually find in some folder you find something like run ICA, remove components. So run ICA, basically all EEG tools have an ICA algorithm implemented. Here I have an example from EEG Lab, but all EEG tools on the market have an ICA. So what does ICA do? Imagine you have three sources in your brain. We're now in the brain. Yeah, not in electrode space at the topography, but now in the head, in the tomography. And we have one source generating this fast oscillation, this green source, bit slow, on the blue, very slow. What you measure on the scalp surface, that's what EEG does. It's always a combination of all signals in the brain. An electrode at the scalp surface never measures just one area in the brain. It's always the sum of all activity patterns in the brain. So if you look at this with 3 electrodes at the scalp surface, then you measure the following. You add up these 3 sine waves, obviously different weighted with different weights, because this green signal is closer to the green, this electrode is closer to the green signal, and so on and so forth. But it's always the sum of all 3. And what the ICA now does is it it will reveal the 3 components, the independent components underlying this summation at the scalp surface. So again, the red, the green, and the blue oscillation. And now you can reconstruct the signal after pruning. Pruning is the word for taking out one component. Let's assume we say the red oscillation is an artifact. We don't want that. So we can take this out and reconstruct the signal. We prune the red IC, the red independent component, and then we end up at the scalp surface with this, so that the artifact is gone now, when we assume the red one is an artifact. So that's what we want to do with the saccadic potential. But the question is, how do we find the independent component related to the SP, the saccadic potential? If you have ever used independent component analysis, you might come across pictures like that. Each component is characterized by a topography, and now you or an algorithm has to decide which component is related to a blink, to an eye movement, to a muscle artifact, whatever. Or in our case, to a saccadic potential. And let me tell you, it's impossible to find that. It's not, one is not able to say, okay, this component number 5, this is the saccadic potential. It doesn't work like that. We have to tell the IC, the independent component, what to look for, so to speak. And how can we tell an independent component analysis what to look for? One idea is we simply add a pattern, a virtual channel which contains the saccadic potential. So just look, ICA, this is what we're looking for. This is how a saccadic potential looks. That's what we want to find. We want to know, you independent component, that this is a saccadic potential. Please look for that. Find me a component exactly for this signal. So now the only problem to solve is, how can we tell the independent component analysis that we're exactly looking for that? If we want to tell the independent component analysis that we're looking for that, we have to find it ourselves. But how do we find it? That's what I was talking about. It's tricky to find it, but there is one trick to find it. As you remember, these saccadic potentials, they are characterized, that's why they are so mean, by their steep slope. Like the Dirac pulse, they're very steep. We can use that to find them. And how do we find the steepness of a curve? You might remember from your math class in the 12th, or I don't know, you learned something about the first gradient, the first derivative of a signal. This gives you the steepness of a curve. So the first derivative gives you the steepness. So how do we find these SPs? They're very small in amplitude. We've used the first derivative because they're characterized by sharp edges. So this is our signal S here. And here we have the first derivative S prime. And here now you see it clearly. The first derivative shows you here at this point in the signal is a very, very sharp edge. That's our saccadic potential. So now we found them if we use the first gradient of our signal. So what we do, we calculate, to summarize the technique, we calculate the first derivative, the first gradient of the signal, best at frontal sensors because it's an artifact stemming from the eyes. Then we detect the maxima in this first derivative, and these are then the saccadic potentials. We set all values which do not belong to the saccadic potential to 0. So we have a pattern, and we take this pattern and hand it over to the independent component analysis and tell the independent component analysis, look for that. And then we simply prune. I told you what pruning is. We prune the independent component which is related to our virtual channel or which is related to our pattern. And then, so the idea, the problem with the microsaccades is gone. That's the idea in theory, in theory. So now I skipped this slide. I just show you at the end here one application of this co-strap algorithm. I want to show you one study, a study in which it worked, obviously. So what we have done, you remember this study, the familiar and unfamiliar picture, which we have published in 2006, and we found higher gamma band responses for familiar as opposed to unfamiliar pictures. And we found this gamma band candle here, which most probably is not a gamma band oscillations, but as we know now, just microsaccades. So we replicated this study. So let's try this one again, we thought. So we have recorded the EEG after presentation of familiar and unfamiliar, and what we found at frontal sensor, the typical gamma candle Same at parietal or posterior sensors, the typical gamma candle before the correction with our algorithm co-strap. And now let's apply this algorithm which we have developed, and wow, it worked really nice. Wow, that's what we thought. I hope I have a little wow feeling in you as well. I don't know. At frontal sensor, the gamma candle is completely gone. There's nothing anymore. At posterior sensors, here in the right lower corner, at posterior sensors, we don't see the gamma band candle at 300 milliseconds anymore, but we see an ongoing gamma band response starting approximately at 200 milliseconds after stimulus onset, ongoing right to the end of stimulus presentation. And if we even look at the sources, I don't want to go into details how source analysis works, but yeah, it's basically a tomography. Before, I would imagine you know how to read these pictures. It's like an fMRI picture also, yeah, like an SPM. It's basically an SPM based on EEG data, a statistical parametric map. And before we use our algorithm, you see activity basically in the eyes. So it's really eye movements. But after our correction, the center of gravity of our gamma band response lies in the inferior temporal cortex in both hemispheres. And that's good. It's not in the eyes, and it's in the inferior temporal cortex. That's where you would expect the location of an object representation to be in your brain. This is your what path of information processing. So we were quite pleased that we, in this study, have dealt with this miniature saccade problem. And then we have obviously also checked, does it still differentiate between familiar and unfamiliar pictures? And yes, it does. It's still higher for familiar. This is as opposed to unfamiliar objects. This is the two red lines here in this graph. The black lines are before correction and the red lines are after correction. So to conclude, the activation of object representation is mirrored in the gamma band response, but also the miniature saccades. They're also higher for familiar as opposed to unfamiliar. So what might happen, a tentative model, what might have happened in previous study is this. So you have here, when you look at the schematic time by frequency plot, you have here this candle-like thing in the gamma band, but that's not real gamma, that's fake gamma. It's miniature saccades. But you also have a real gamma, a real cortical oscillation, which is an ongoing oscillation in the frequency range, which is not stretched out across all frequencies, but it occurs in a specific frequency range. And at this point in time, around 300 milliseconds, the fake gamma and the real gamma, they have an overlap. That is what might happen. So now we have also replicated some of our memory studies, repetition priming. I just skipped these slides and just let me tell you it works. So we still have sharpening and formation even after correcting for miniature saccades. With a look on my watch, I skipped these slides, and also a very recent study where we have applied this algorithm in a virtual reality setup. It even works in virtual reality. I come directly to my conclusions. Object representations, as mirrored by induced gamma band responses, are no fixed entities. They're influenced by learning and memory. But unfortunately, this also holds for microsaccades. It seems as if they're also modulated by learning and memory. However, the claim brought up by Yuval Greenberg in 2008 that all induced gamma band responses in the EEG can be fully explained by microsaccade-related fluctuations. This does, in my view, not hold. There is also real gamma oscillations in the human EEG. By application of appropriate data preparation, it is possible to examine induced gamma band responses during various cognitive processes. So I would say if you're into EEG research, keep your filters open. It's not necessary to filter your EEG at 20 hertz because you say, oh, we don't see the gamma anyway. No, no. Keep your filters open. It is feasible to measure gamma in the EEG. It's not easy, but it can be done. So having said that, I like to thank you very much for your attention. And on the left side, you see the names of all my collaborators. I thank them as well. And as I said, you for your attention. Thank you.
Thank you very much. That was a quite interesting topic. And I mean, now it is almost the time you need to leave, as you mentioned before.
Yeah, sorry.
But it is about quarter past. Might there be time for one more question, or would you like to leave early today?
No. I have another 10 minutes or so. It's rather a question. You did send me questions. I can answer these questions, or I can take questions from the audience as you wish. I just that there were quite interesting questions, and I did like, especially, yeah, the first one was good. There was a question, maybe I just take this one from the list you did send me. There was a question, why did gamma band receive such a hype and not other frequency bands? And I think this is, I thought about it, and I really, in my opinion, it's not the frequency band which received the hype, but it was really the topic, this binding problem. This is quite, in my view, it's worth a hype. I mean, in neuroscience, you have kind of a continuum, more boring things to answer. I mean, we have to be honest, there's research when people look at attention and then they check if the P1 component changes in amplitude if you switch your stimulus from red to green. Yeah, it's interesting, maybe, but to be honest, it's a bit boring. And all at the other end of the continuum, you have consciousness. How does consciousness work? And gamma band the binding problem is kind of in between, bit more towards consciousness. So really an interesting topic. And then there was this finding that has to do with gamma band responses. And that's my view. That's why I think it was such a hype. It was really a hype, yeah. But yeah, the other, if I have answered all your questions in writing, I can send you, I will send you my slides, and I can send you obviously the answers to all the questions that were sent in. So you have then all the answers. But now, if there are questions in the audience, please feel free to ask.
From what I see, there is no question until now in particular, but I have one that is more or less related to one of the questions I sent here. So we had a presentation about neuromodulation as a treatment for Alzheimer's disease, where we were shown results that steady-state response can increase memory, especially high frequencies, so frequencies that would relate to gamma band activity. But if you then combine the steady-state response with gamma-regarded oscillation research, where would you say you can do the cutoff between the confounding of those two effects? Because at some point it might be also there's an ongoing discussion we were told about if those induced oscillations actually affect cognitive processes. And in this result, it seems a little bit like, but overall, it tends more towards, there's no real affection of cognitive process. What is your perspective on that, and how would you handle this problem?
I mean, obviously, it's a very straightforward idea, and we had this idea. If I was talking about the theta response related to episodic encoding. So we assumed, okay, if we flicker our stimuli at 7 hertz, we should see better encoding. We should facilitate encoding. So we tried that. We flickered at 7 hertz, we flickered at 10 hertz, which is then in the alpha range, usually a sign of relaxation, maybe not the best frequency for memory encoding. And we not really flickered in the gamma band range, but we also presented static stimuli. And if you assume that a monitor runs at 60 hertz, then we also presented stimuli in the gamma frequency range. They were static, but they flickered at 60 hertz. Our brain perceives this 60 hertz in V1, V2, but it's not a conscious, it's above flicker fusion frequency. You see a static picture. And what we found is really that we flicker in the theta range, then memory is better than when we flicker in the alpha range. But memory is still the best for a static picture. And now I don't know if this is due to the fact that a static picture is a 60 hertz picture, or it's just a static picture. I don't know. But at least there is something. So if you flicker at 7 hertz, memory is better than at 10 hertz. And then best when you flicker at 60 hertz or have a static pictures, there is something. But I think there's a lot of this. It was only small effects. And this is because, at least in my view, this is why it's something that is hard to do research on, because everybody has his gamma frequency or his theta frequency is inter-individually very different. The one has it at 7, the other at 8 hertz. Your gamma might be at 40 hertz, mine at 50. So first I have to find out what is your theta response. Then your theta response, how would I know in an experiment at what cycle you are at the moment of the stimulus presentation? Stimulus presentation. So I need to record and stimulate at the same time. And now you see it gets really, really tricky. Maybe if somebody has a lot of time and doesn't have to do research on the safe side, it has to be tested. But as far as I know, nobody has tested it yet in its best form.
Thank you very much. So there's one more question.
If we have time for that, then I would be really thankful. At first, thank you for the interesting presentation. It was a nice overview over the last 20 years nearly. I have a question regarding the differentiation between beta and gamma. So depending on the definition, gamma activity can start as low as 25 hertz, as you also showed in your presentation that Gammaband activity is defined as. So there's an overlap between high beta and low gamma activity. And I was wondering if it could be, if there could be any potential value in the distinction between high beta and low beta, especially when looking for differences in functional connectivity and functional connectivity networks in emotion processing. I know it's a really specific question, but I would be really interested in what you think about that.
I mean, what I think is quite clear that this another long-held belief that beta responses are simply responses from the motor cortex, I think that is not true. You also have this high beta band responses, which are somehow related to memory as well. And then you have the low gamma responses, which overlap. And here I think it's a definition. I mean, if I say it's high beta or low gamma, I would say it doesn't matter. You can call it whatever you like. The problem is more if you say it's a high beta or a low gamma. It doesn't matter. But what is the, I would rather ask the question, what is then the difference between a low gamma and a high gamma? And here it gets tricky because some researchers claim, yeah, the lower frequencies are more the large scale integration of large scale networks. And the higher you get in frequency, the more focal are the networks which have to be integrated. But then I look at MEG research, and basically, I would assume with MEG you measure similar networks as you do with EEG, but in MEG you measure higher frequencies if you look at gamma. That makes no sense to me. So here I really have to say it is. I think it's that to come to a point. It doesn't matter if you call it high gamma, high low gamma or high beta. 25. It's a 25 hertz response. Point. It doesn't need a name. I think it's more interesting how to differentiate it from the high gamma. Is it really the case that you have here a difference in local or focal, and what is local and focal? I don't have an answer to that yet.
Okay.
So
Thank
sorry,
you.
I have no answer to your question.
No, of course, it's no problem. Um, I know that it's. I had some insight into papers about beta and research that tries to differentiate exactly that between low beta and high beta. Um, they also draw the connection that high beta might be associated with large scale networks and stabilization in functional connectivity networks, whereas the low beta might be more associated with the motor cortex or whatever. It's really unclear at the moment. I know that. And since I'm doing a project with some face processing data, EEG data at the moment. I was interested if it could be valuable to dissociate between, especially within beta and high beta and low beta too, and not only be just look at beta as one frequency and average over it because of this overlap with the low gamma frequency.
I mean, one heuristic might be what helped me. If I look at a time by frequency plot, and you say, for example, yeah, you have a low beta, and I look at the time by frequency plot, and I don't see a distinct blob in the low beta, but it's just a smearing out of the upper alpha band, then I would say, okay, that a beta? It's no, it's just a smearing out of the upper alpha. There is absolutely nothing. So if I want to see a specific band, as I said, the name doesn't matter, but I need a blob in the time by frequency a plot. If there's no blob, then it's not a distinct signal, but just a smearing out of another frequency. And then there's, yeah, it might be an overinterpretation of upper alpha.
Yes, that could be true. Thank you.
But I would say it's a modest time for you to leave. Thank you very much for having us for a presentation, and I wish you a very nice evening. We will come to the ending slides, but you have a nice evening, and see you next week.
Thank you again for your attention. Good night.
So for those remaining, I will just take over and do our normal ending slides. Second. So that was it. Thank you for being here until now. And as usual, if you want to see more from us, just look up our website and social media stuff. The next Neuro Talk evening is on the 20th of October, so like around about 2 weeks, or exactly 2 weeks. And then it is about the TIPS study, which is a multimodal neuroimaging study where multiple researchers from the University of Münster contributed to, and they will give us a group presentation, what might be pretty interesting. Also, if you want to look up Professor Gruber's research, you can, of course, look it up on his website. Actually, he doesn't have a Google Scholar, I know for a fact. So therefore, this is the only option to get some more insights. But yeah, thank you very much and have a nice evening. See ya.